Okay, good morning, everyone. So what I'd like to start with today is the bonus question from last time, which was, if we have somebody who has a deficiency in Bruton's tyrosine kinase, which is the kinase immediately downstream of the signaling proteins of the B-cell receptor, namely the Ig alpha and Ig beta, where would the cells arrest, or where would they get stuck? And if you think about it, during T-cell and B-cell development, as we'll see for T-cells in a moment, it is rearrangement of the genes during somatic recombination, and then test for if some protein is being expressed on the cell surface. So the first test of that is whether or not the heavy chain was rearranged productively or in frame, and that's the transition from pro-B to pre-B. And so if you don't have that signaling, the B cell never knows that it has a heavy chain on the surface. And so B negative SCID, which is severe combined immunodeficiency patients that lack B cells, Bruton's tyrosine kinase is the most common mutation. So if you were to look at all of these all of the B cells that are developing in the bone marrow, they would be all stuck at this pro-B stage, and they would never go to the large pre-B proliferative phase. Now, Bruton tyrosine kinase is important for going from the pre-B to immature B cell stage, but they never get there. And so they'd all be stuck before that. Okay, are there any questions on that before we move ahead?
So in Janeway's book, because they had said there that it gets stuck at the pre-B cell to the immature B cell when they were talking about specifically Bruton's tyrosine kinase?
It's incorrect. They get stuck, they never make it to the pre-B stage.
Okay.
Now there are other kinases like Blink that can compensate for that, and so biology is never quite as clean as we make it in the lecture. And so there are some ways that you can get sort of aberrant pre-B cells, but then they also get stuck at the next stage, transition to immature B cell. But in reality, most of them are stuck at the pro-B cell stage. Okay, so today we're going to move from the B cell receptor to the T cell receptor. And the T cell receptor is a little different in how it recognizes antigen, and so it has a different structure. And so we have to go back to how did we know that T cells do anything? And really, we didn't know what T cells did. If you remember, in the early part of the 20th century, it was thought that lymphocytes were really not very important cells because they were small, they didn't have much cytoplasm, they were often called unremarkable lymphocytes, and people thought that they were on their way to senescence and death. But one guy that really thought that maybe these cells had something different was that he observed that if you had in animal models or in human disease, that the cells that always infiltrated into tuberculin masses or cancer, or if you did tissue transplant and the tissue is being rejected, the cells that infiltrated those tissues were always lymphocytes. And so this was the first indication that lymphocytes were important. But by and large, it was mostly just rejected because people didn't think they did much. And so what he did was this experiment. And remember, this is before the advent of tissue culture. And so instead of a petri dish or a tissue culture flask, they would use embryonated chicken eggs as the vessel to test things. And so what they would do, or what he did, is he would take tumors or tissue from one strain of mouse, okay, and put it in the chicken egg. And then he would take lymphocytes or lymph node cells from another mouse, put them in the same chicken egg, and then see, is that tissue or tumor rejected? Does it get killed? And the answer was, yeah. And so this was proof that yes, lymphocytes that you get from a lymph node actually do something. Now, we didn't know how they did it, and we didn't know which lymphocytes were important, but it was the first proof that lymphocytes actually did something. Okay, and that really came down to these guys, George Snell and Peter Gorer. And these are the guys that really started Jackson Laboratories, where we get most of our genetically uh, modified mice or inbred strains of mice. And what they did is they genetically mapped the histocompatibility antigens. Histocompatibility just means tissue compatibility. And they called these the H antigens and showed that these were responsible for tissue rejection. Now, at the time, this was pre genome sequencing. So what they were doing was genetic linkage, showing that these sets of genes that they mapped by how close are they to other genes were responsible for this. And so this is, if we go back to genetics, this is sort of the centimorgan measure of, it's not actually base pairs, it's more about how close are they. And so what they do, take two strains of mice and breed them together. And they would test the F1, does that accept the tissue from the first strain of mouse? And then they would breed that strain again back to strain B and keep doing that. And at each stage, they would test, did that offspring accept a graft from the initial parent? Okay, and when they did, they could say, well, this was this many generations, and so this is how closely the genes map. And by doing this in many strains of mice, they could determine that these H antigens were the genes that were responsible. Now remember, this historically, we didn't really know what genes were. We didn't really know how the DNA was encoding for proteins. This time we knew that DNA was probably the genetic material, right? But we didn't really understand how it got to the actual traits that they're measuring. Okay, so in this kind of experiment, what you're measuring is how many times do you have to breed those mice back to each other in order to get acceptance of the donor's skin transplant? Or in this case, how many times do you have to breed them together to get rejection? And that really brought us to these guys who you've seen before, right? That measured that T cells still develop in chickens lacking the bursa fabricus, but not in thymectomized chickens. And so if you remember, they used either salmonella infection or tissue grafts to look at B cells versus T cell responses. And so if they thymectomized chickens, the chickens would not reject skin grafts from unrelated donors, meaning that T cells were responsible for this tissue rejection. And then, of course, Graham Mitchell and Jock Miller, who sort of repeated all of the experiments that Robert Good and Max Cooper did, but did it in mice. And in sublethally irradiated mice, they showed that if you thymectomized those mice, they would not reject tumors. And so the experiment looks like this. You take a mouse and you irradiate it. That irradiation removes all of the bone marrow derived cells. Okay, it kills off all of your hematopoietic cells. You then take bone marrow and put it back into that mouse. Now, if you do that, that mouse will then reject a skin graft from a different strain of mice. But if you thymectomize the mice, you give it back bone marrow, but you thymectomize it, it will accept the grafts or not reject them because there's no T cells. Okay, so this was the proof that in chickens and in mice that the thymus was necessary to reject skin grafts from an unrelated donor, which said that lymphocytes are the ones that are doing this. Now, at that time, the problem was that we didn't have monoclonal antibodies to identify lymphocytes. We didn't have ways of growing lymphocytes, and so we needed new tools. And it really came down to Lloyd Old and Edward Boyce. Lloyd Old is the pioneer or the grandfather of tumor antigens. He's responsible for identifying most of the tumor antigens that we know about. But they developed antibodies against two different proteins. At the time, they were called L3T4, which is, we now know is the CD4 protein, and an antibody against the LIGHT2 antigen, which is lytic 2 antigen, which identified CD8. Okay, so now they had tools to start isolating T cells and say which T cells are important. And so the way that they did this is they would take thymus cells from a mouse, or thymus tumor cells in this case, and they would put them into a rat. Now the rat will make antibodies against the antigens that it's never seen before. So it's going to make antibodies against the mouse proteins that are expressed in the thymus. They would then go on to create hybridomas, which we talked about what a hybridoma is. And those hybridomas produce monoclonal antibodies, and they screen these for which ones react with mouse thymus cells. And they found these antibodies against CD4 and CD8. And that was all well and good, but we couldn't really do much in tissue culture with these because you still had to be able to grow the cells. And so that really ended up being what people did before this is they would use conditioned media. So they would take CD4 cells, stimulate them with mitogens so that they make something, and they made T cell growth factor. Well, they didn't know what that was. So Kendall Smith, who was at the time at Dartmouth, identified and cloned the T cell growth factor gene, which we now call interleukin 2. This is a T cell stimulating gene that allows these cells to proliferate. And so now you could isolate the cells and you could grow them up in the laboratory and start analyzing what did they do. So with these tools, Mark Davis and Steve Hedrick at Stanford, Steve's now at UC Santa Barbara, and Tak Mak at University of Toronto both did this experiment where they used subtractive hybridization to clone mouse and human TCR genes. And this experiment really was a landmark because it had never been done before. How do you figure out which genes are important? And they made an inherent assumption. The assumption was that essentially B cells and T cells, they're both lymphocytes, so they should have, all the other genes should be the same, but except for the B cell receptor or the T cell receptor. And so what they did is they took B cells and T cells from mice, they made mRNA, and then they reverse transcribed the mRNA from T cells into cDNA. So now this cDNA is complementary, is the reverse complement of any mRNA that is the same between the two cells. Okay. Now, of course, at this time, we didn't have fancy ways of labeling DNA, so they actually did this in P32 radio labeled nucleotides so that they could run this on a gel and just identify bands that were the same. Now, the trick here is that you hybridize these together. So you form DNA, RNA complexes whenever the genes are the same between the two cells. Okay, now if you form these double-stranded hybrid complexes, those will bind to hydroxyapatite. Hydroxyapatite is essentially kitty litter. It's a naturally occurring version of kitty litter. And double-stranded RNA, DNA complexes will bind to it, but single-stranded will not. And so the single-stranded DNA was still free in solution. If it's single-stranded, it is unique to the T cell. B cells don't express it, and so they're looking for things that are unique to T cells but don't occur in B cells. So they would run these on a gel, cut them out, and then use sequencing to identify what those genes are. Okay. So the inherent assumption is that the only thing different between T cells and B cells is the receptor. Now, in reality, we know that that's nonsense, right? T cells and B cells have very different jobs. There's lots of other proteins that are, or genes that are different between these two. And so it's kind of a flawed assumption, but they got lucky. They found that one of the 15 genes that they isolated looked a lot like an antibody, but was unique to the T cell receptor and had a very different structure. In reality, Mark Davis and Steve Hedrick, who usually get credit for this, are the ones that actually identified the TCR gamma gene, whereas TACMAC identified TCR alpha genes. And so they both should get credit. They did it about the same time. Mark and Steve published this a little earlier, so they generally are the ones who get credit for this. There were also others at the time that were doing similar things, but they didn't publish it first. So that's the way that science goes. So what did they find? So if you look at the genes encoding the T cell receptor, they looked a lot like the FAB fragment of an antibody. They had a variable region and a constant region. They had an alpha chain and a beta chain, so instead of heavy and light, you have alpha and beta, because they're roughly the same size. And they have a transmembrane domain. They also had the same sort of structure in that they were immunoglobulin taco domains, right? These were beta pleated sheets that were folded over and stapled with a cysteine bond. So they essentially look just like the FAB part of an antibody, but they functioned a little differently. And some of the differences are that if you look at the DNA sequence, there's no secretion version of this. It is only membrane bound. What they also noticed is that unlike antibodies, there's no isotype differences. All of the TCR just have the same constant region. There's no IgM versus IgG. There's no isotypes. Okay, so T cell receptors seem to be a little more static. They don't change as much as a B cell receptor. And we'll talk more about that when we go into B cell and T cell activation, where B cells diversify at the receptor level, where T cells diversify at the cellular level. And that's because they have very different roles or very different functions in the immune system. So when we look at these domains, we come back to this idea of the immunoglobulin taco, right? They're essentially beta pleated sheets that are folded over and stapled with the cysteine bond. What we also notice is just like the antibody FABs, the antigen binding parts are just these fingers extending from the beta pleated sheets. Okay, so it looks just like an antibody in terms of the structure. So for a T cell receptor, it has 3 hypervariable regions, or 3 complementarity determining regions, 3 for the alpha and 3 for the beta. And those occur at these loops at the ends of the fingers on the taco. And so the structure looks very much like an FAB. So if you have the V alpha and C alpha for TCR alpha, you can see that those are both immunoglobulin domains, and the V beta and C beta for the TCR beta are also the immunoglobulin domains. And you'll notice that all 6 of the CDR regions are just at the tip of the TCR, just like what we see for an FAB fragment of an antibody. Okay, now, as I said, TACMAC identified the TCR alpha, and Mark Davis and Steve Hedrick identified the delta chain. And what that result of that was that there are actually 2 different types of T cell receptor. We'll go into that a little bit more, but For the most part in this class, we're just going to talk about the alpha beta T cell receptor because those are your conventional T cells. Gamma delta T cells have a sort of different role in the immune system. They are more like innate cells, just hiding out in the periphery, waiting for things to respond to. They have much less diversity in the terms of the recombination. So we don't go a lot into the gamma delta T cells. The main ones that we're going to focus on are the alpha beta T cells, which are conventional. So the alpha beta recognize classical MHC proteins, MHC1 or MHC2, that are presenting peptides that are derived from either extracellular or intracellular sources. These are what we'll focus on. The gamma delta T cells recognize either intact structures like a B cell receptor would, or they recognize non-classical MHC proteins that are presenting glycolipids or lipids that are on peptides. And it's really too much for this class to go into those cells. But we do cover them in the advanced course if you're interested in following up on that. So if we go back to an antibody or a B cell receptor, the B cell receptor does not have any inherent signaling domains. And this was sort of a problem of how do you get B cell activation if there's no signaling proteins or signaling domains for the B cell receptor. And that's solved by pairing those with the Ig alpha and Ig beta. These have ITAM domains that allow a signal to a B cell. It's not really a conformational change. People argued about this for a long time. I can remember even in graduate school, people didn't know how a B cell receptor signaled. It's more grouping all of the ITAMs together to overcome any phosphatase activity that would limit their function. T cells have much lower affinity for antigen. So they do have this antigen binding part on their T cell receptor, but there's no signaling domains. And because the interaction is much weaker, you actually have many more signaling proteins. So you have the CD3 proteins, there's epsilon, gamma, delta, and zeta. Now of these, it turns out that zeta is the most important. The other CD3 proteins can provide some signal, and it's either epsilon and gamma or delta and epsilon pairs, but the CD3 proteins have the most intracellular tyrosine activation domains, and so those are the ones that are most important for T cell activation. But if you just compare, Ig alpha and beta have 2, whereas the T cell receptor has 10. So there's many more signaling domains for the T cell receptor. But just like for the B cell receptor, you have to have T cells that recognize everything. And so the same logic applies that you can't just encode a T cell receptor gene for any potential thing coming in. You have to use somatic recombination. And so if you look at the TCR loci, it looks a lot like the B cell receptor loci, where you have variable diversity and joining segments. And there's one small difference, and that is that the delta loci actually occurs inside of the alpha loci. So here's the delta. Oops, should have glued this part. That's the TCR delta gene segments. And you have flanking that the TCR alpha gene segments. But in any case, what happens is you're going to get rearrangement between an alpha V and an alpha J. When that happens, you end up cutting this part out. Remember, you get this excision circle that says in the cytoplasm and it gets degraded. For the TCR beta, it's essentially the same as the heavy chain. You have a D to J rearrangement, and then you have a V to D to J rearrangement. And that gives you a productively arranged TCR sequence. Now, just like for the B cell receptor, as those genes are rearranged, that brings the promoter closer to the enhancer and turns on mRNA expression. And so it shouldn't surprise you that TCR recombination follows the exact same rules as B cell receptor recombination. You have control of it by the RSSs, the recombination signal sequences. And so again, you have the 7 nucleotide with a 23 spacer followed by a 9 nucleotide sequence. Then you have a 9 nucleotide followed by a 12 nucleotide spacer and a 7 nucleotide repeat. And that's so that you can pair up the 7 nucleotides, pair up with this 7 nucleotides, these 9 nucleotides, pair up with these 9 nucleotides, and what you get is a mismatch. You get a bulge in the DNA on one side, and that allows the RAG1 protein to recognize, yes, there's something paired up here, we can rejoin these. Okay, so only gene segments with a 23 nucleotide RSS can recombine with gene segments that have a 12 nucleotide RSS. And this is the exact same rule for T cells and B cells. There's one small difference, okay? So for the light chain, it looks just like the IgA kappa or Ig lambda, where you just have 23 RSSs with the V gene segments and 12 RSSs for the J gene segments. There's one hitch. For the TCR beta, the J's have 12s, the V's have 23, and the D's have a 12 on one side and a 23 on the other side. So in theory, you would join D to J with this 1223, and then V to D to J with this 12 and 23. But there's a problem, or the idea is that you can actually join this 23 RSS to this 12 RSS. Remember, though, there's multiple D gene segments, right? There's one here, one here. This is in humans, but in mice, there's multiple of these. So the idea is that you could actually get several D gene segments in the same TCR structure. Now, in reality, that doesn't happen very much, and it's very difficult to prove because exonucleases will come in and they'll start chewing away those regions. The Ds are very small, and so often the Ds get obliterated during somatic recombination. But in any case, the same rules apply, right? You have to have a 23 RSS with a 12 RSS. Okay, so let's come back to the TCR alpha. And one of the ways that the T cell defines whether it's going to be a TCR alpha beta T cell or TCR gamma delta T cell is whether or not you get TCR delta gene rearrangement. And this happens before TCR alpha rearrangement. So you get basically this shot of, okay, I can make gamma delta T cells. If there's no delta rearrangement, then it proceeds to the alpha stage or to alpha rearrangement. So what happens is there is a difference in which gene segments rearrange first. And based on what you know about the B cell receptor rearrangement, which TCR genes are going to rearrange first? Anybody got a guess for that? Okay, so it may not be intuitive, but it breaks down to beta, gamma, and delta all rearrange first. And this is to give you a chance to say, is it going to be an alpha beta TCR or a gamma delta TCR? And it occurs the same way that you have for the B cell receptor. Okay, so you get TCR, delta, gamma, and beta all rearranged first. It turns on somatic recombination of all of them at the same time. Now, if you think about it, to get a TCR gamma and delta, you need both chains in frame, rearranged, and then that cell could become a gamma delta TCR. But that's, you know, a 33% chance times 33% chance. So it's really only 11% chance you'll get both of those together. The TCR beta, on the other hand, only has to rearrange one. So it's very similar to the heavy chain. If you get a beta rearrangement, then that works. And so that's about 33% of the time that's what's going to happen first. So really you get about 3-to-1 alpha beta versus gamma delta. Okay? And that really determines what kind of cell is it going to become. And there's differences in how those cells mature. The second step is you're going to have TCR alpha rearrangement. So if you get a beta on the surface, then you will rearrange the alpha chain, just like the BCR light chain. Okay, so we're going to go through somatic recombination one more time, just because it's not as complicated as people initially, when they first see this, think, oh my gosh, this is so complicated, but it's not really. It's the same steps, just for a different receptor. Okay, so you have these lymphocyte unique proteins, and the main ones are RAG1 and 2, terminal deoxynucleotide transferase, and Artemis. And so the first step is the same as for a B-cell receptor. The RAG proteins bind to the RSSs. You form this, this complex, and the second step is that RAG2 will cut the DNA. That releases the signal joint, or the signal sequence, and you have the coding sequence. For the coding sequence, you get covalently closed ends of the DNA, just like you do for the heavy chain and light chain rearrangement. Whereas for the cut out signal joints, it's free DNA ends. Now, of course, the next step is you have these proteins, Ku70 and 80, come in and bind to ends of those cut DNA. And it's exactly the same steps for the signal sequence. You know, the Ku70, 80 proteins are bound to those ends. DNA ligase comes in and closes the ends, and you get this excision circle. Okay? And that, again, can be used to identify recently matured T cells. They've just come out of the thymus. They haven't had a chance to degrade that excision circle, and so they still are young T cells. Now, one difference here is that when this happens for the alpha chain of the T cell receptor, that cuts out the entire delta loci. And so you force the T cell into not being able to become a gamma delta T cell if it rearranges the alpha chain. And then just like the B cell receptor, most of the time, those genes are in the same orientation, meaning the 23 RSS is facing the 12 RSS. And so when you get that, you get the excision circle. But if they're pointing in the same direction, meaning the 23 RSS is pointing away from the 12 RSS, then you get this gene inversion. For the purposes of this class, the main thing that happens is they're pointing towards each other, which you can think of as an opposite orientation, and that will result in an excision circle. And that's the main way that this happens. There are actually very few gene segments that are in the opposite orientation. Okay, now let's talk about the coding sequence. The exact same thing is happening for T cell receptors as for B cell receptors. DNA PKCS is going to come in and activate Artemis. Artemis is then going to cleave that hairpin at some random site to give you, is it P nucleotides or N nucleotides? Anybody?
P nucleotides?
Right. It's going to give you a palindromic sequence because it's cleaving a hairpin. And so then once that folds out, it's going to be read the exact same way forwards and backwards. What's the next step? Well, just like for the B cell receptor, terminal deoxynucleotide transferase is going to come in and add non-templated nucleotides to the ends of that cleaved DNA. Remember, the non-templated means N nucleotides, okay? And then you're going to get micropairing, and the exonucleases will cleave the excess parts that are not pairing with the other chain. The DNA repair enzymes come in and fill the unpaired nucleotides, and then DNA ligase 4 seals that joint. Okay, so just like for a B cell receptor, you have the same process of making sort of a sloppy joint, and so you get lots of different sequences. So the coding sequence has an imprecise joint, meaning you've added a bunch of things, you've created randomness at that joint. The signal sequence you don't care about, so you just seal it up and get rid of it, so it has a precise joint. So we look at this at the nucleotide level, right? We have our two things are brought together due to their RSSs, and RAG complex will cut those, and the signal sequence then forms these hairpins, right? Here and here. And so when Artemis comes in and cleaves that at some space, you get this palindromic sequence. So it's TCGA, but the complement of that would be TCGA. And so that's the palindrome part. Then you get TDT is going to add some non-templated nucleotides, right? And then those will start to get these micro pairing here, where they start to pair up. So then you have these parts that are sort of flapping in the breeze, and exonucleases will come in and cut those off. You then get the polymerases that fill in these gaps. And then DNA ligase 4 seals those gaps. And you're left with a complete sequence of joined together. So I've made a quiz in Canvas, and I'd like you all to open that now. Or you should be able to open that now. Okay, and this is going to be an in-class exercise. There's no points on this, but if anybody gets 100% on this quiz, everyone will get 100% on the final. Okay, and we're going to make T-cell receptors. And I actually have, this is not something we normally do in person with pipe cleaners and little pieces of felt, but we're going to try and do this virtually. Okay, so is everybody able to open that quiz?
Yeah.
Okay. So let's say you're going to make a TCR beta. You have to choose a D and J segment, right? So just like the heavy chain, it's first D to J. So for the first two questions, you have to pick, we're just going to use colors for these, okay? You have to pick a D and a J gene segment colors. And guarantee you that the colors that are on the screen now are not necessarily the ones that I've picked. I actually have real ones that I've made that are sitting in front of me now. I'll make sure not turn on the camera so everybody sees. Okay, so you have to pick a D and a J gene segment. Okay, the next step is you have to now choose how many N nucleotides are you going to make there. We're just going to sort of skip over the P nucleotides and say, okay, you got to choose. You can make between 1 and 4 N nucleotides between the D and the J gene segment. And again, we're just going to use colors for these. You realize that I have 6 possible ones there. But you have to choose, is it going to be 1 nucleotide long or is it going to be 4 nucleotides long? And then for each of those, you'll need to choose which color of nucleotide it's going to be. Everybody with me so far? Why don't you give me a thumbs up if you are at that point?
I'm sorry, would you mind just repeating instructions one more time?
Sure.
Thank you so much.
Okay, so you've chosen a color for the D gene segment, just saying which one did you choose, and a color for the J gene segment. Now you have to choose, TDT is going to come in and add nucleotides between those two. And you get to determine, you're going to say how many it adds, between 1 and 4 nucleotides.
Sorry, I had a question here. So you asked us to choose between 1 to 4 nucleotides. We can choose, for example, 2 and pick up the color. So for the questions that ask us the color of the third or the fourth, we just skip those questions. That's
If you
right.
notice at the bottom, it says none. So if you chose 2, then your color for your third and fourth would be none.
Mm-hmm. Okay, thank you.
All right. Everybody there? Okay, so let's keep going. Now you have to choose a V gene segment for the TCR beta. The next question is, what color is that? And then
So I
just
believe that's question 8, right?
correct. I'm not actually loaded into Canvas right now. I'm actually just in the slides.
No worries. I think I'm trying to follow along.
So the question is, how do you determine the color of the end nucleotides? You get to pick. The goal here is you want to come up with the same T cell receptor that I have.
Has anyone ever gotten 100% or gotten your colors exactly?
There's always a chance. Not so far. All right, now, just like the other joint, you're going to choose between 1 and 4 connector or N nucleotides to join the V to the D to J. And for each of those that you choose, you have to choose what color it's going to be. And if you choose 3 nucleotides, as I have in this example, the color for the 4th one would be none, because there's nothing there.
So that's going to be questions 9 through 13.
Am I seeing that color correctly? Because that last ring is red.
Well, these are not the ones that I've chosen.
Oh, okay.
These are just an example. Otherwise, I'd just be giving you all the cheat sheet for the final, right? Okay, would you give me a thumbs up or raise your hand if you're all the way there?
Should we go on to building the alpha V segment?
Yeah, that's the next thing is now you have the TCR beta. Now you got to make a TCR alpha. So again, you're going to have to choose a V and J gene segment and tell me what color those are going to be. And they're also going to choose the N nucleotides. How many N nucleotides are going to put there and what are the colors? And so those should be the last questions. And once you have your TCR sequence, everything inputted just the way you like, hit submit for the quiz. And also raise your hand or give me a thumbs up in Zoom. Now again, the more of you that try this, the more chances there are that everyone in the class will get 100% on the final. We'll give you just a couple minutes to do that. The people who have logged on or dozed off will be sorry they didn't get a chance. I see some of you are seeing the math problem behind this. Okay, is there anybody who still needs time?
Yes, I do. My computer froze. Just give me a second, please.
Same here, please.
Seeing a lot of 6 out of 20.
I
Oh,
think the
there's no,
highest right now is 10 out of 20.
there's no retakes. All right, I'm gonna go ahead and close it so we can keep moving on. And you'll see why I can be so confident in, um, in saying that we're not going to be able to get a correct answer. Evelyn, can you confirm for me that it's actually unpublished now?
Yeah, give me a sec. Yes, it's unpublished.
Okay. So I am going to show you my TCR.
It's kind of hard to see.
Professor, I think
You're
you
muted.
muted yourself.
Here's my A beta chain. Okay, so it's obviously not the ones that were on the PowerPoint slide. This is what you would normally make in person. Okay, and just so you know that I'm being honest, here's my alpha chain with my V and my J, and then my N nucleotides. In this case, I picked 3. So nobody got 100% on it. And why do I know that? Well, if we go back to our numbers here, and somebody was pretty close in the chat about what our possibilities are. So if you just look at the number of different TCR beta that are possible here, you only have 5 colors. And in reality, there's something like 140 V beta gene segments. And there's roughly 12 J gene segments, and then a couple D gene segments. But when you multiply the possibilities together, you actually get quite high. So there's, in this case, 5 different colors. You can have 4 different lengths of N nucleotides at that joint, but there were 7 choices. And then you have 5 colors for the D gene segments, roughly the same number of nucleotides at that joint, and then 5 colors for the J. When you get to the TCR alpha, you have sort of the same calculations, but the alpha is much less diverse than the beta just because you only have 1 joint instead of 2. Now, in reality, the complexity of the T cell receptor rearrangement itself is much, much higher than this because you obviously have more gene segments. You can have a much higher number of N nucleotides that are added, but just your chances here, right, are something like 1 in 43 trillion. Okay, so to get this level of diversity, you see that the junctional nucleotides are the main factor for increasing the total diversity. If it was just which colors did you pick for V, D, and J, you'd have about 1 in 3,000 chance. But once you start adding in nucleotides, it becomes 1 in 43 trillion. Consider how many people are on the planet, right? We're roughly about 7 billion. That means if we had, I don't know, 400 and something different Earths, and we had everybody on the planet do this, maybe somebody would come up with the right answer, but it's extremely diverse population. So the whole point of this exercise is to show you that the N nucleotides are the main thing that's driving TCR and B cell receptor diversity. Now, I wish that we could have done this in person because it's more fun to have something in your hand. We're actually making these, but I hope this was at least a partially fun exercise. Okay, are there any questions on that before we go on? All right. Okay, so now the question is, you've generated a diverse population of T cells with all different kinds of T cell receptors. We still don't know how they actually recognize stuff. And so that really came down to the people who are generally given credit for this are Rolf Zinkerlegel and Peter Doherty, but again, there were others doing very similar stuff. Peter was at St. Jude's in Tennessee, but he's also faculty in Australia. Rolf was his postdoc working in his lab. And in the late 70s, they showed that T cells have to recognize both a self protein and a foreign protein. But others, Mike Bevan at the University of Washington and Tak Mak at Toronto, showed essentially the same thing. Okay, and so this is what they did. They infected mice with a virus, in this case, lymphocytic choriomeningitis virus. And this mouse had the MHC-A haplotype, and they knew that all this means is all the MHC are an A type. Okay, and they knew that from Peter Gore and George Snell's experiments that these were inbred mice, and so they could use this to test, do the T cells that they isolate, right? The T cells are now, Lloyd Old had isolated antibodies, so they isolated CD8 T cells from these mice, and they say, okay, if I have MHC-A cells, so the same MHC as the host, do the T cells kill those cells even though there's no virus? So there's no virus, but the right MHC, they don't kill. If they have the wrong MHC, but they do have virus, they still don't kill. You need virus there and the right MHC. Now, Rolf and Peter got the Nobel Prize for this in 1996, saying that this is how T cells recognize things. In reality, they were wrong. They proposed that T cells have two receptors, one for the self and one for the foreign virus. This ultimately is incorrect. It's the same receptor that's recognizing both. Okay, so even though they got it wrong, still got the Nobel Prize because they were mostly right and sort of figured it out later. So the way that this works is that T cell receptors are recognizing the self protein, in this case, the MHC or major histocompatibility complex genes that Peter Gore and George Snell defined in their inbred mouse experiments, and the foreign virus peptide, or the bit of the virus that's recognized. If you have the wrong MHC, T cells can't bind, okay? They just don't recognize it. If you have the right MHC, but the wrong peptide is being displayed, you also don't get recognition. Okay, so this really showed that you have to have both TCR and MHC. And it really came down to, I guess I've left that slide out, to figuring out how the T cells recognize this came down to crystal structures of this. And there were a number of people who did this. And it was really showing that the antigen is a peptide that is bound in the groove of an MHC protein. Okay, so you might notice that parts of the MHC protein are immunoglobulin tacos. So here's one, beta-pleated sheets that are folded over with a cysteine-cysteine bond. Of the MHC1, there's a second protein called beta-2 microglobulin, which is also an Ig domain protein, beta-pleated sheets folded over with the cysteine-cysteine staple. But the actual parts of the MHC that present the peptide to the T cells are a little different. They do have some beta-pleated sheets, but if you notice, the alpha-1 and alpha-2 domains of the MHC1 or actually have these alpha helices, okay, that are sort of the sides of the presenting part of the antigen. And the peptide lays in the middle of it. And for me, it's easiest, I know I use a lot of food reference, this is no longer a taco, this is a hot dog bun, okay, where the peptide is sitting in between the two edges of the MHC alpha helices. Okay, now there's two different MHCs, right? We remember that CD8 T cells will recognize MHC class 1, and CD4 T cells will recognize MHC class 2, and they have a lot of structural similarity, but there's some differences in where they're expressed, and that's a lot to do with what their role is. So MHC1 is important for CD8 T cells to recognize. Well, what is the job of CD8 T cells? It's to kill any virally infected cell. So virtually every cell in your body will express MHC class 1, and this even includes neurons, particularly if they're in an inflammation setting. Now for MHC class 1, there is a single MHC protein, okay? It has alpha 1, alpha 2, and alpha 3 domains, and then there's a soluble protein that binds with it called the beta 2 microglobulin, okay? For MHC2, it again has this alpha helices that are on the sides of the peptide binding groove, but instead of having one MHC protein with a beta 2 microglobulin, there's a MHC class 2 alpha chain and an MHC2 class 2 beta chain. And so the domains are actually named the same way. The furthest one out is the lowest number. If you remember, for the antibodies, the constant domain, the constant region domains are numbered. The farthest one out is the lowest number. Okay, so the numbering is the same for both of them, but don't get confused. It's basically an alpha and a beta chain for the MHC2, and an alpha chain with beta 2 microglobulin for the MHC1. Now MHC2 is not expressed everywhere. If you think about the job of CD4 T cells, it's to activate other immune cells and control the response. So you really want that only on professional antigen presenting cells, and so it's limited expression, just on the cells that are going to be important for CD4 T cell activation. Okay, so now we have to talk about anchors. Excuse me. How do these MHC bind peptides? So if you all remember from biochemistry, where you have these amino acid bonds, essentially every other amino acid side chain is facing away from each other, right? So if you were to stretch out a peptide, the first amino acid side chain is going to be pointing down, the second one will be pointing up, the third one will be pointing down, the fourth one will be pointing up, and so on. That's just the way that the peptides are structured. Now, the immune system takes advantage of this and says, okay, the ones that are pointing down, those are going to be anchor residues. That's what's going to bind to the MHC. And the parts that are pointing up are going to be the TCR contact residues. That's the part that the T cell receptor is going to see. So a TCR, or T cell receptor, is going to bind parts of the MHC on those alpha helices. And then it will bind to parts of the peptide, or the TCR contact amino acid side chains that are pointing up out of that MHC. Okay, so you have to, the T cell recognizes both. Now, it's a little bit different. T cell receptor recognition is a little bit different than B cell receptor. Turns out for T cells, the most important contacts are actually the hydrophobic residues buried inside of that peptide binding groove. So there's sort of the outside parts are the ionic bond bridges with that it's recognized as the MHC, but it's the hydrophobic residues of the peptide that are actually being exposed. So MHC genes are actually interesting in that they're codominantly expressed, and you have many different versions of these in the population. MHC is the most polymorphic gene in any species, or any mammalian species. It is, so if you were to look at anyone else in your class, it's unlikely that any two people in this course have the same set of MHC genes, unless you happen to be related. I said that one year and there were identical twins, and so I had to eat my words, right? And so you have, for MHC1, you have 3 MHC1 genes. You have HLA-A, HLA-B, and HLA-C. HLA stands for human leukocyte antigen. In mice, you have mice also have 3 MHC1 genes, and they're just called H2D, H2K, or H2L. okay? Now, that's polygenic or polygyny, where you have many copies of the same gene that essentially serve the same function. So polygyny is multiple copies of the same protein. But these genes are also polymorphic in that you have many different versions. So you have many different versions of HLA-A, many different versions of HLA-B, and many different versions of HLA-C. So that's polymorphism. Now, these are all codominantly expressed. So you actually have 6 copies of HLA. You have 2 A's, 2 B's, and 2 C's. They're all expressed. And so my MHC type, I know because I worked in a research lab, is I have HLA-A2 or HLA-A02. I have HLA-A22, HLA-B14, HLA-B44, HLA-C08, and HLA-C18. Okay, so that's my MHC, and that determines what my T cells are going to be able to recognize. It's unlikely that any of you would have that same set, because we're not related, and the number of different versions of those genes is so high in the population, it's very unlikely that we would have those. Now the difference is, inbred mice are bred so that they have the same sets of MHC. And so we often denote that in shorthand. And this shorthand only applies to inbred strains of mice. You don't have this in humans. So C57 black 6 mice, which was very common in the lab, have H2B, which means that both of their versions of H2D are D of B, and both of their versions of K are H2K of B. So we often just note that as shorthand as H2B. In C57 black 6 mice, they've actually lost the L gene due to the inbreeding. They've lost so they only have 2. BALB/C mice also have lost a gene. They've lost the D gene, and so they have just 2 MHC1. They have H2L of D and H2K of D, and that means both copies of L are L of D, and both copies of K are K of D. So in humans we use numbers, in mice we use superscript. Now the shorthand for BALB/C mice is their H2D, so meaning all of their alleles are the D haplotype. Okay, now we can go into this for MHC2, where you have 3 MHC2s. In humans we call them HLA-DP, DQ, and DR. And when we go into MHC processing, you'll see there's some others that help with the loading of presentation of these, but essentially they have 3, or humans all have 3 of these, but because you have 2 sets of those genes, you actually have 6 different MHC2. In mice, most inbred strains of mice actually only have 2 MHC2. They have H2IA and H2IE. So if I were to ask you in black 6 mice that are expressing H2IA, what is the H2IA haplotype or allele? Anybody can answer. Well, I haven't told you what it is, but look up here. They're H2B. So what do you expect the IA allele to be? It's going to be IA of B, right? So all of the MHC genes are the B haplotype. If I were to ask you in BALB/C mice, what is the IE gene? It would be IE of D, because they are H2D. Okay, so when you look at, we said that MHC are the most polymorphic proteins or genes that you express. You look, where do the polymorphisms occur? Well, most of them occur right at the tip of this peptide hot dog, right? Where they're occurring in the bun. And they can occur either in the sides of the bun, the part that the T cell receptor is going to see, or they can occur in the bottom of the bun, which is what determines what anchor residues that MHC is going to bind, okay? So this is, the MHC polymorphisms are affecting which peptides are going to be able to present it to T cells. So if you're looking at the peptides from a virus-infected black 6 mouse that are being recognized by T cells, they're totally different than the peptides that are recognized by T cells from a BALB/c mouse. Now, when you look at the MHC1 protein, the two parts that are most variable are the alpha 1 and alpha 2 domains. That's because that's the part that binds to the peptide, right? This is the alpha 1 domain, this is the alpha 2 domain. You see most variability is in those two domains. The alpha 3 doesn't bind peptide, it's just structural, so it's essentially the same as a constant region domain. And it's the least variable, and that's the part that CD8 alpha binds to. Okay, so if we look at, this is actually a crystal structure from David Garbozzi, who was the first to crystallize a TCR bound to an MHC and peptide, which is a very difficult thing because it's not a terribly high interaction or high affinity interaction. But essentially he got all of these in a soluble form, crystallized it, and was able to get a crystal structure. And if you just look at the part that's binding here, okay, so we're, the alpha helices are on each side, the peptide here is in yellow, and then you have the CDR1, 2, and 3 from the T cell receptor. And you just look at overlay where those occur. Here's the CDR1, 2, and 3 for the alpha chain. And here's the CDR1, 2, and 3 for the TCR beta chain. Now, the CDR3s are the parts that are in yellow, okay? And so the CDR1 and 2 parts of the T cell receptor make major contacts with the MHC, right? Those are the parts that are binding to the beta helices on the outside of the hot dog. The CDR3 is the part that makes major contact with the actual peptide that's bound inside of that groove. So the CDR3 is recognizing the hot dog, the CDR1 and 2 are recognizing the bun. And so that's how a T cell receptor can recognize both at the same time. Now you have the same interactions for TCR as BCR, whereas you have ionic bonds and hydrogen bonds. Those are true for the outside when it's binding the bun, but the actual peptide in the middle, the hot dog, is actually hydrophobic side chains are the most important because that determines whether or not the TCR is going to get activated. Okay, so John Altman, who also worked with Mark Davis, developed something called a very unique tool called the MHC tetramer. And TCR MHC interactions are very low affinity, and so they don't really work like an antibody. But if you increase the number of binding sites, you increase avidity, even though it's a low affinity thing. Okay, and so if you remember this experiment where I showed you, oh, I got LCMV, and then they analyzed my T cells, and I got this huge proportion of them, both CD4 and CD8 T cells, are recognizing the virus. Well, how do we do that? These are actually MHC tetramers, and so they're MHC1, here's a beta-2 microglobulin, and these have a biotin label on them. Biotin is bound strongly by streptavidin, and so it makes it into a tetrahedral shape. And so essentially you can have 3 to 4 arms binding at any one time to the T cell receptor, and so then you can identify antigen-specific T cells. Now this was really a new tool. John Altman developed this right around, he developed in the mid-90s, and it sort of came to fruition around 97, 98, and was the first time you could identify antigen-specific T cells without a functional assay. And so that's what I'm showing down here. Here's an MHC1, HLA-A2, with a peptide from the virus. Here's an HLA MHC class 2 that's binding the peptide from a different part of the virus. And these will only label antigen-specific T cells. Now, the second part of MHC binding is you have these co-receptors, right? And so you have CD4 and CD8. CD4 for CD4 T cells and CD8 for CD8 T cells. And they have different structures, but essentially they're made up of Ig domains, beta-pleated sheets folded over with a cysteine-cysteine bond. And their job is to bind to MHC in the non-polymorphic regions. CD4 is monomeric, CD8 is heterodimeric, it can be it's dimeric, can be hetero or homodimers, but essentially they're binding to the conserved parts of MHC proteins. And their job is to increase antigen binding. So CD4 is going to bind to the beta 2 chain of the MHC and stabilize the TCR interactions. CD8 is going to bind to the alpha 3 domain to stabilize CD8 TCR interactions with MHC1. Somebody's got something going on there. Okay, so if you don't have a question, please mute yourself. I'm getting some interesting sounds. Okay.
You're muted.
Rolf and Peter showed that you need the right MHC and the virus to be there, right? So that was, you need the right MHC and they don't kill cells with a different MHC. But this whole time, we've been talking about rejection of tissues based on MHC. So if it's the wrong MHC, it gets rejected. And that is a problem, is that some percentage of your cells are alloreactive, meaning they will automatically react with a MHC that they haven't been trained on before. And so when we talk about T cell development, in positive selection, they have to be recognizing the right MHC. But now you introduce a foreign MHC, some percentage of those might actually recognize that foreign MHC, and they don't care what peptide is there. Okay, so alloreactivity is when a T cell recognizes some foreign MHC independent of peptides. And there is a peptide there, but the T cells are just recognizing the foreign MHC. And it's basically a TCR doesn't care what the peptide is. It's just binding so strongly to that foreign MHC protein that it gets activated. And bacteria often will take advantage of this by expressing something called a superantigen. A superantigen forces a TCR to interact with the MHC protein, and the reason it does this is that it just activates T cells non-specifically, they go on to die, and the bacteria can go on to cause more disease or replicate, okay? So this is sort of nature imitating what we see in the lab, is superantigens. Okay, so I realize we've gone a few minutes over. What do I want you to know? I want you to know how TCR gene segments are rearranged. It's the exact same process as a B cell rearrangement with this added issue of you can get either alpha, beta, or gamma delta, but not both. And again, how does this relate to getting one TCR per cell and the clonal selection theory? And then what are the steps? And sort of, I think we've skipped over a little bit of this. The pre-TCR, I think we're gonna cover again during T cell development in the next lecture. Okay, so the difference, you should know the differences between a TCR and a BCR FAB, and then how the the way T cells recognize peptides bound to MHC proteins. And then you should be able to define what polymorphism and polygyny are. So That leads into the next bonus question, which will be up in Canvas in a few minutes, is why do we have polymorphic and polygenic MHC proteins? Why is that good for us as a species or for an individual? And you can answer either at a species level or an individual level on Canvas. And so with that, I will stop and take questions. So yes, so I didn't really highlight it, but I said, as we talked about earlier in the semester, CD8 cells recognize MHC class 1, so 1 times 8 is 8. CD4 cells recognize class 2, so 4 times 2 is 8. That's how you remember which one recognizes which. Okay, so is there any questions? Okay, if there's no more questions, I will see you on Thursday.